going to be a big picture overview of anemia. So how did we divide anemias? We divide anemias by their red blood cell size, so the MCV. We have the microcytic, normocytic, and then macrocytic anemias. So microcytic anemias, what was the MCV? MCV, if you see MCV less than 80, you're talking about microcytic anemias. And there's a couple of them, and we're going to use iron studies to differentiate. And what was the problem? What was the common cause that leads to microcytic anemia? And the common problem is hemoglobin synthesis. And how did we get hemoglobin? Remember that hemoglobin is made up of heme and then globin. So if you have problems synthesizing either of these, you're going to get a microcytic anemia. So heme synthesis, globin synthesis. So what were problems in heme synthesis? Remember that heme was made out of porphyrins plus iron. So if you have a problem in either iron or porphyrin production, you're going to get heme synthesis. So things like iron deficiency anemia and anemia of chronic disease cause decreased iron. And remember that sideroblastic anemia and lead poisoning cause decreased porphyrin production. Remember there was problems in the enzymes along that pathway that I showed you. So that's it for the problems in heme synthesis. So iron deficiency anemia and anemia of chronic disease, and then for the porphyrins you get sideroblastic anemia, lead poisoning, and then problems in globin synthesis. What did I tell you? When you have the alpha and the beta thalassemias. Remember, these were congenital defects, inherited defects in the genes that were needed for beta chain and alpha chain production. And remember, you can have the alpha thalassemia minor or major, or there's the 4 genes in alpha thalassemia with the spectrum of disease, and then beta thalassemia has beta thalassemia minor and beta thalassemia major. So, let's erase this. Let's go on to normocytic anemia. What was the MCV here? Remember, MCV was from 80 to 100. That's a normocytic anemia. How did we divide normocytic anemias? Remember, we divided it whether it was hemolytic or non-hemolytic. That was the big dividing factor. And how do we tell which one was which? You saw MCV between 80 to 100. How do you know it's non-hemolytic versus hemolytic? Remember, the answer was you look at the reticulocyte count. And in hemolytic anemia, what happens to the reticulocyte count? Remember, it's going to be increased because you're going to be replenishing those hemolyzed red blood cells, so you're going to have over 2% reticulocytes. Non-hemolytic, remember the problem here, what's the problem? Is inability to make red blood cells. So you're going to have decreased reticulocytes as well, so it's going to be less than 2%. So hemolytic, remember hemolytic we can divide even more. We can divide it, the way I taught you was intrinsic versus extrinsic problems. So intrinsic to the red blood cell causing hemolysis, extrinsic causing hemolysis. Remember the other way we can divide it is whether the hemolysis occurs extravascularly, that is in the liver and the spleen, or whether it occurs intravascularly in the blood vessels. And then intravascular versus extravascular is going to have different lab findings. Now let's go back to the way we learned it, intrinsic versus extrinsic problems. And then intrinsic problems I divide even more. There's membrane defects, enzyme deficiencies, and hemoglobin mutations. That's the big picture. So what were the membrane defects that you could see that causes intrinsic problems? There were 2 membrane defects you could see. You could see one was paroxysmal nocturnal hemoglobinuria, PNH, and the other was hereditary spherocytosis. Remember, PNH was the underlying problem here. What was missing? Remember, the missing thing was the GPI anchor that you need for DAF, decay accelerating factor. Remember, and DAF is important because it helps get rid of complement, helps inactivate complement. So if you're missing that, you have too much complement activation, you get the hemolysis. The other problem was this hereditary spherocytosis. What was the problem here? What was the missing stuff? Where the missing stuff here was you're missing the proteins that anchor the red blood cell membrane to the cytoskeletons. You're missing ankyrin, band 3, spectrin. And then because of that, you get the blebbing, you get fragile red blood cells. Remember, it's fragile, that's the key word there. Alright, so the enzyme deficiencies. What were the 2 enzyme deficiencies that we talked about? We talked about G6PD deficiency, and we talked about pyruvate kinase deficiency. And why was G6PD important? Remember, G6PD, we need to regenerate reduced glutathione, which helps get rid of the oxidative damage. And then remember, what precipitated the hemolytic crises in G6PD deficiency? Remember, it's ingestion of things that increase oxidative stress. So things like the anti-malarials, things like fava beans, having an infection. I think Bactrim sulfate drugs was another one. So that can all cause the hemolytic, the oxidative stress, and a hemolytic crisis. So pyruvate kinase deficiency, this was a shorter one that we didn't talk too much about. But pyruvate, remember red blood cells, how do we use the energy? Remember, red blood cells have to use glucose, and they have to be able to break down that glucose, and you need pyruvate kinase to do so. So if you're deficient in pyruvate kinase, you can't break down glucose, you're going to have problems in the red blood cells. Finally, hemoglobin mutations. Remember, we have the two hemoglobin mutations, which were sickle cell disease and hemoglobin C disease. For sickle cell disease, you get mutation of hemoglobin A to hemoglobin S. Do you remember the specific mutation? Where it's glutamic acid to valine. And so you're going to have a lot of problems, which I talked about. And then hemoglobin C, remember, was actually, let's go back to sickle cell. What were the big picture problems? Remember, big picture problems where you get hemolysis, you get severe anemia from hemolysis, and then you're going to get the occlusion of blood vessels from the sickled cells, and the occlusion is going to cause problems all over the body, including the lungs. That's acute chest syndrome. You're going to have problems in the vessels for the bones of the hands and the feet. Remember, that's the dactylitis. You can have problems in the kidney. You get papillary necrosis, all parts of the body. And the hemoglobin C, remember, is just a more mild version of sickle cell anemia. Remember the mutation here. Again, it's glutamic acid mutation, but it turns into lysine instead. Remember that big L. Let's go back. Excuse me. Remember lysine. It was glutamic acid to lysine, and we remember that because of that lysine. Okay. So that's it for the intrinsic problems, intrinsic to the red blood cell. Now we're going to talk about extrinsic problems. So what were the extrinsic causes of hemolytic anemia? Remember, the extrinsic causes included that autoimmune hemolytic anemia with the warm and cold. Then you had the microangiopathic anemias, which we talked about, so TTP, HUS, and you can also have DIC, that's consumption. You get clots, and platelet clots, and the red blood cells get sheared. You can also get shearing from macroangiopathic anemia, so that's in large vessels. And what were the two risk factors for that? The two risk factors that I mentioned were having prosthetic valves or having an aortic stenosis that causes shearing. Finally, infection from malaria and babesia, because these infections go into they like to grow in the red blood cells, and they cause rupture of the red blood cells. So that's it for the hemolytic anemias. Now we shift to the non-hemolytic anemias. And so the two non-hemolytic anemias I talked about, what were they? Remember, the problem here is not destruction, but failure of production. Failure of production in red blood cells. So anemia of chronic disease, which I told you could also be macrocytic. And remember, what was the problem in anemia of chronic disease? Remember, anemia of chronic disease, you get acute phase reactants, you get too much hepcidin, which basically inhibits iron transport. So you have decreased available iron to be used for red blood cell production. And then aplastic anemia was the other one. It's just a problem in the erythroid progenitor cells. And there was many causes that I told you about. So the causes include infection, radiation, drugs, autoimmune causes impaired production of blood cells in general, actually. Red blood cells, white blood cells, platelets. So that's it for the normocytic anemias. Now we're going to shift gears to the macrocytic anemias. So this is where the MCV is over 100. Remember, what was the big way we separated macrocytic anemias? Remember, we could separate it into whether it was megaloblastic versus whether it was non-megaloblastic. And just going backwards again, macrocytic anemias, what was the problem here? Remember, macrocytic anemias, the problem was in red blood cell production. And so you have a decreased, actually you have too much hemoglobin concentration in red blood cells, so red blood cells are going to become bigger to balance that out. And so megaloblastic, the problem is in impaired DNA synthesis, so you get impaired red blood cell production consequently. And what was the characteristic features of megaloblastic anemias? Where you had the polysegmented neutrophils with over five of the over 5 lobes. What were the causes of megaloblastic anemia? I told you folate deficiency, vitamin B12 deficiency, orotic aciduria. Folate deficiency from diet. What was the diet? Remember, it was the healthy stuff, so fruits and vegetables. Vitamin B deficiency, not from the diet because you have nice big stores of it, but vitamin B12 is from animal products. The deficiency occurs from pernicious anemia or damage to the ileum, so Crohn's disease. So you have either damage to the intrinsic factor, damage to the ileum. And then orotic aciduria is the other one, And then finally, non-megaloblastic anemia. The problem here is impaired red blood cell production. It's not an impaired DNA synthesis, it's some other impairment in red blood cell production. I told you there are 3 causes: alcoholism, liver failure, and finally Diamond-Blackfan anemia. So that's it. That's our whole review of anemias. You don't have to memorize exactly, like, you don't have to memorize what, like, and list out what are all the intrinsic problems of all the intrinsic hemolytic anemias, or what are all the, I don't know, all the non-megaloblastic macrocytic anemias, but be sure to be able to know the pathophysiology and how each of these work, and understand that, let's say, aplastic anemia would be normocytic, folate would be macrocytic, because you're going to see they're going to tell you it's a macrocytic anemia. You're going to have to figure out what's the problem. They're going to tell you it's normocytic, they're going to tell you reticulocytes are low, you're going to have to figure out what's the problem. So that's it for my overview of anemias.